The overall goal of this procedure is to follow endocardial cells during atrioventricular canal and heart valve formation.
This method can help to address key questions in the field of cardiac development, such as to understand how endocardial cells behave in different types of mutants. Tracking cells during AVC and valve formation is challenging because of the rapid beat of the heart. It makes it difficult to follow cells via traditional time-lapse microscopy. The main advantage of this technique is that it bypasses some of these difficulties. Demonstrating the procedure will be Renee, a postdoc in my lab.
After creating a mold according to the text protocol, pipette about 1.5 milliliters of melted 1% agarose into a 35 millimeter plastic mounting dish. Then, position the plastic mold in the liquid agarose, taking care to avoid trapping air between the mold and the agarose. Place the dish at 4 degrees Celsius until the agarose hardens. Then remove the plastic mold from the agarose. Next, add 2 milliliters of previously prepared mounting medium to the mounting dish and allow 15 to 30 minutes for the solution to diffuse into the agarose. In the meantime, melt a 1 milliliter aliquot of 0.7% low melting point or LMP agarose by placing the tube in a 70 degrees Celsius heat block for about 5 minutes.